Our next speaker is Dr. Jared Miller, and Dr. Miller grew up on a farm in Harford County and has degrees in environmental science and soil science from Virginia Tech and the University of Kentucky. He has a background in soil formation, nutrient cycling, and carbon sequestration. He has been an extension educator in Somerset County since 2014. Today, Dr. Miller will be presenting his presentation titled Soil pH and Lime Rate. Welcome, Dr. Miller.
All right, so this is to talk about soil pH and lime rate that I know some people that come to some of these agronomy days may have already seen it. This will be the third time I've given this one in about 24 hours. But down here in the lower shore we have a large variability in soil types, which has got me interested in being able to discuss the benefits of lime, understanding lime rate and soil types and how they're all tied together and affected. So first off, we want to start by discussing with people and yourself that what are the benefits of lime and why do we lime soils in the first place. The number one reason would be to neutralize acidity, to raise the pH of your soil. But part of neutralizing that acidity and raising your pH can also reduce toxic metals like aluminum and manganese. It can, depending on your source of lime, add calcium and magnesium. Another thing that might be overlooked is that the soil biology, just like your crops, is going to prefer a certain pH, and that soil biology itself helps in nutrient cycling. So if you can, okay, we have poor audio. Okay, I'll back up just a little bit. Calcium, magnesium can come from lime. They are a great source if you need them in your soil. Another thing about lime in your soil and raising that pH is your soil biology likes to have a certain pH range itself, because you have to remember these microbes are in there helping cycle your nutrients. Just like your crops, microbes and soil biology are like a certain pH range. They're not going to prefer an acidic pH in most cases for what we want our crops to do. And then also some pesticides are going to prefer a certain pH range. So there's a lot of benefits to liming. It's not just neutralizing acidity. It's a good thing, and I know there's always a range of knowledge in any audience you go to make sure you understand what pH is. So pure water is H2O. It's hydrogen and hydroxyls. The hydrogen is the proton. It's that positive charge. So the hydrogen is the acid. The hydroxyl, the OH, is the base. So if you increase the amount of hydrogen, which could be by adding hydrochloric acid, you make a solution more acidic. If you add more hydroxyls, you make the solution more basic or alkaline. So that P, the pH, is actually a measure of the concentration of hydrogen. But if you notice, as your pH goes down, you have more acidity. So it's good to get an understanding of what that P stands for. The P means negative log base 10, and I'm not trying to make you refer back to math, but the log base 10 allows us to take things that change in a, I say, large manner in the case of pH, and get it on a scale that you can view. So in this case, when you're going from a pH of 5 to 6, it's actually a 10 times change. That's what that log base 10 does. So when you go from a pH of 5 to 6, you actually have 10 times less hydrogen. And if you look over to the left of the graph, you can see how small these concentrations actually are, so that at pH of 6, you're actually at a concentration of 10 to the negative 6 hydrogen ions. And that's why they stick that log in there, so we can have positive numbers, and it makes more sense when you discuss it. But pH drops because of this. The hydrogen concentration drops as your pH goes down. In soils, what causes acidity? Hydrogen is the acid. So hydrogen is the main thing to talk about and understand and discuss, but aluminum as a metal can cause hydrolysis, which is the splitting of water. And when it splits water, it creates more hydrogen, so it creates acid. Iron can to a small extent. Calcium, magnesium, potassium, sodium, we consider those bases because they don't split water. Since they don't split water, they don't create acidity, so we can call those the bases. So in your soil, in your lab test reports, Hydrogen and aluminum represent acidity in soil. Calcium, magnesium, potassium, sodium all represent the bases. Aluminum and iron can be related to soil mineralogy and weathering, how much aluminum iron you have in your soil. And you got to remember that hydrogen can come from manure and fertilizer, so we can have different sources of those acids. Water itself, pure water with no ions in it, has a pH of 7. That means it has an even amount of hydrogen and hydroxyls. But carbon dioxide in the atmosphere actually dissolves in water and creates carbonic acid. It lowers the pH of water. So if you look over on the left side of this diagram, right about here where we have natural rain has a pH that's less than 6. So water in the environment doesn't have a pH of 7 because of the ions that can be in it. So carbon dioxide actually lowers the pH of water. If you look over to the right side of all our different soil types and where they fit within pH range, you have the humid region arable soils. We're in the humid region because we get more rainfall than other parts of the country. So one way I explain this to people is if you think about our soils, if they're washed over thousands and thousands of years with slightly acidic rainwater, then you could also expect our soils to be slightly acidic. And that's why most of the soils we deal with sit between about a pH of 5 and 7. If you go out to the western part of the country, you can have more calcareous and sodic soils where we have less evaporation and you can have salts build up. You can find calcareous soils. I've seen it in some limestone soils. If you happen to be near acid mine drainage or some reduced sulfur that can oxidize, you can have even more acidic soils. So our soils are going to be in our region naturally acidic, partially because of weathering, but you can relate it to how they are washed with a acidic rainfall. So what causes these soils to be acidic? A lot of it can be due to weathering. Minerals in our soils can contain a lot of aluminum, and as they break down in weather over time, they're going to release that aluminum. So the older your soil is, the more weathering that's occurred, you can have a lot more aluminum, and it means the soil can be more acidic in its nature. So on the left side here, we have what you consider an environment to be warm and wet, a climate like this, where you have plenty of rainfall, leaching your soils, and elements like sodium, which has a charge of 1, potassium, magnesium, calcium, they're held looser in the soil than aluminum. So sodium leaches out first, followed by potassium, magnesium, calcium. So over time, if you don't add these bases back in, your soil builds up in aluminum. So in an environment like we are on the East Coast, if you have soils that are very old, that are highly weathered, they can be more acidic because they're going to be built up in aluminum, versus a cooler climate where if you have more evaporation, that means you can't wash the salts out, and you actually can gather salts on the surface of some of these soils. On the left here we have a map of soil types, and on the right you have a map of precipitation. And this kind of goes back to the point we're talking about how weathering and rainfall influence the soil types and also influence acidity. So if you look over at the soil map, this orange here are the ultisols. These are highly weathered old soils, high in clay, high in iron and aluminum. As you move across the country, you can notice that it matches the decrease in rainfall. So climate has a large effect. It's one of our 5 soil forming factors. Climate has a large effect on the soil types we have, and it's also going to affect how acidic they are. So we have old weathered soils here with high rainfall, and we have these desert soils that might be higher in salts because they don't wash out calcium, magnesium, sodium. But you'll also notice down here this little path where we have the Appalachian Mountains. So soils are also controlled, the type of soil you have. Parent material is another soil forming factor, and it's another factor that can affect the type of acidity you might have. So if you look at the state of Maryland, if you cut across Maryland, we have the Allegheny Plateau, the Valley and the Blue Ridge, Piedmont, and then the coastal plain region. So a soil over here, depending on the parent material, because we can have sandstones and shales, but you can also have limestone in this region. In some of these soils, if you have enough, you can have carbonates, you can have higher pH in these soils. So a region over here, we can actually have soils naturally have a higher pH. The Piedmont region, where we have soils weathering from bedrock, they might be older, they might be more acidic depending on how weathered they are, but one thing about soils that have bedrock is this rock, even though it's weathering slowly, can be adding nutrients back to the soil, depending on the type of rock it is. So mineral weathering can keep these soils replenished with micronutrients, with other nutrients, even though they might be older compared to the coastal plain. And the coastal plain can be very variable, but all these soils in this region of Maryland are going to be sediments that washed off of this region. So since all of our bases, if they're dissolved in water, are a lot easier to carry out to the ocean, and clay and silt and things that have better CEC and nutrient holding are easier to carry to the ocean, our soils can be dominated by sandy textures that are low in nutrients. And the coastal plain is variable. We can have higher clayey regions. We can have organic matter. It doesn't mean necessarily that your soil is going to be automatically lower in nutrients, but if you look across the region and you do relative comparisons, you could say that these soils should be lower in nutrients and have more hard-to-weather silica-type minerals. Well, these might have replenishment in nutrients, and you could even have some higher bases here. So relative in the region, you can see some differences. So pH is simply a measure of acidity. Why does your soil test report have 2 pHs? Well, the first pH, it should just be labeled pH, is what we call the active acidity. The active acidity is what your crops see, it's what your biology sees. It's a very important measurement to let you know whether or not you want to raise the pH. So if you like to have a pH of 6.5 for your fields, this pH tells you you're too low. The buffer pH is one that tells us how much additional acidity can be held in reserve on the soil. Buffer pH is important for when we're making lime additions. It doesn't necessarily tell you how acidic the soil is. It tells you how much acid is held in reserve so that when we want to lime these soils, how much additional lime you'll have to add. So the first pH is very important in understanding what pH do you want to be at for crop growth, for biology, for your pesticides. And the second one lets us know if you're going to lime to raise this pH, you might have to add more lime than you would think. So, why do we need a buffer pH? That regular pH, the active pH, you only measure the acidity dissolved in the water. So you take a soil sample, and it can be in a 1-to-1 ratio. You take 1 gram of soil, and you can also dissolve and have 1 gram of water, a simple mixture, and the pH probe goes in there and measures the acidity. The buffer pH is a way that we look at the acidity that's held in reserve, what's absorbed to the soil, so that if you add lime to get rid of this acid, is there going to be additional acid that comes off the surface of the soil? Now, buffer pH can get complex. If you're talking about it in a soils course, you can talk about residual acidity and pH-dependent acidity. But usually when I'm giving this presentation to farmers, when we're talking about agriculture, because we sit in pHs that range from about 5 to 7, exchangeable is probably going to dominate what controls our reserve acidity, which would be your CEC. So, most people know this, but I always include it for farmers just in case. CEC helps explain reserve acidity, but you should understand what CEC is. I use this same slide in almost every single talk I give. Soils have negative charges, mostly. Clay and organic matter can have negative charges. Many of our soil nutrients, calcium, magnesium, potassium, sodium, are all positively charged, so those are the cations and cation exchange capacity. So are acids, hydrogen and aluminum, are positively charged. So they're attracted to soil and they can exchange on and off. So the larger your CEC, the more acids and bases you can hold. So that's how that will contribute to your buffer capacity or your buffer pH. Buffer pH is a measurement we do in the lab, but buffer capacity is how much that soil is buffered, how much reserve acidity or how many bases it can hold. So, in this case again, usually discussing soils for agriculture, sitting between about a pH of 5 to 7. It's mostly going to be related to CEC to simplify the discussion. So soils with a greater CEC have a greater buffer capacity, and this is actually a good thing. Sometimes when you're discussing this and you become apparent, if you have greater buffer capacity, it means I have to add more lime. But what that buffer does, it's just like having a buffer along the stream. It slows a rapid pH change, either up or down. So the greater capacity your soil has to either hold acids or bases means it's not going to change as rapidly if a more acidic or more basic rainfall or water, lime, manure, or fertilizer is added to the soil. You got to think about microbes and crops just like us. They're not going to want a rapid change in pH. So if your soil is well buffered, you might have to add more lime, but it also means it can acidify slower, and you're not going to have rapid changes in pH. Base saturation refers to, and if you try to do the math on these elements, they don't add up to the CEC, but it's easier to see them if there's even numbers on there. Base saturation refers to how many bases are absorbed to the cation exchange capacity. So think about our bases being calcium, magnesium, sodium, and potassium, and then aluminum and hydrogen are our acids. So if you have a soil with 50% base saturation, half of its bases, half of its acids, 75% means 75% of its bases. So anything that has a lower base saturation should also have a lower pH. So this is where buffer capacity becomes important and why you can't add the same amount of lime to each soil just depending on the pH they have. So you can have two soils that have, say, 50% base saturation. They both have a similar pH, and you're going to want to raise that pH. The first soil, if you add the same amount of lime to each soil, you can quickly oversaturate one that has a low buffer capacity. And this is a reason that we do buffer pH, and it's a reason that we do lime recommendations, because if you oversaturate a soil, if you raise the pH too high, which you'll see later, we get a lot of additional problems with losing access to micronutrients. So the actual buffer capacity is very important when considering how much lime to add. There can also be a variation in CEC with pH. This is a slide that I leave out for farmers, but I'm definitely putting it back in for you guys, because there's been some discussion with me sometimes that I have raised my pH by adding lime, so therefore I'm going to lime my soil up to a pH of 8 because it increases my CEC, and that's great for my soil. But there's a reason your CEC is increasing. You're not actually changing it in the soil with pH. If you look over here on the left, you see this is a graph of whole soil, which means sand, silt, clay, organic matter, whatever in there. And on the bottom, we have a pH that raises from about 3 to 8. And then you have the CEC. These soils down here that already have a pretty low CEC, as you raise the pH, so does their CEC. This is called pH-dependent charge. These ones with a higher CEC initially also have a larger change as you get above about a pH of 5. So this is a rapid change. A lot of this can probably be tied to actually organic matter, because if you look over here, when we separate clay from organic matter, as you increase the pH with clay, clay has a moderate increase in CEC. Organic matter has a large increase. So soils with a high buffer capacity, it might have a lot of organic matter or maybe even a lot of clay. As you increase the pH, that also means you can increase the buffer capacity to a certain degree. So buffer capacity isn't exactly constant in all soils. This is a diagram from good old Brady and Weil. I haven't updated my copy since 2001, but this is why I tell most people, if you look between about a pH of 5 to 7.5, buffer capacity is dominated by cation exchange within this range. So when I discuss this with farmers and I talk with people about agriculture, I know that I can just say, focus on your CEC, and you'll understand how well buffered your soil is. But your soil can be buffered by carbonates at high pHs, and it can be buffered by aluminum compounds at lower pHs. So if you've had a soil science course, you might have a better understanding of this. But in most cases, we don't want our pH to drop below 5, and we certainly don't want it this high. So if regular pH is just, say, a 1-to-1 mixture of 10 milliliters of water to 10 grams of soil, buffer pH, you take a soil and you stick it in a buffered solution. In this case, it will have an alkaline pH. It could be 7, 7.5, 8. In this case, we have one that's a pH of about 8. If you put the soil in there and you leave it to equilibrate over a certain amount of time, any additional acidity that's on the soil is going to come off and lower this pH. So if you come back the next day and this pH is only about 7.9, you know you didn't have a lot of reserve acidity. If it drops to 6.5, it gives us an idea of how much additional acid was absorbed to the soil. So this helps us calculate how much lime you'll need if you want to raise your pH. The thing is, from what we've discussed about soil types and weathering and climate and parent material, you know that not all soils are the same. Not all soils have the same CEC, the same organic matter. So you can't measure the same buffer pH the same way on all soils. Because of this, there have been a lot of different tests that have been created and calibrated across the country, and they're picked to match what they're going to assume is the average soil in the region. So this is a diagram. It was 1996, looking at what were some of the dominant soil tests used across the country. So you can see one is called the SMP, done throughout the Midwest, Pennsylvania, some up in the Northeast. Adams-Evans, done by the University of Delaware. Maryland is actually listed as other because back then, my understanding is they used soil texture as part of their lab, so they had a test as other. The Malick is one that is still done in the state of North Carolina. And this is important because you can send your samples off to several labs that will use different tests. So it's good to understand, and this is what I tell farmers, when you're sending your tests around, to know, do all your soils match the test they're running, and if you run them between different labs, because you don't want to overshoot your lime. You don't want to add too much. So that SMP buffer was meant for soils that have a high lime requirement, but low organic matter, of a content less than 10%. Now, over here in Princess Anne, if we had 10%, that would be considered very high organic matter. We're usually sitting, if we're lucky, between 2 and 4. So already you can see within our region this test might not fit the best over here on the coastal plain. It was based out of soils in Ohio. They would have taken these soils, done calibrations in the lab to figure out what's the buffer pH going to be and how do we relate that to lime requirement. You have to run these different calibrations in a lab to make sure you understand how it works. So you take something like this that's been done over time and is used across the country in different labs. If they're not recalibrated and retested, you can't always be sure how accurate they are. In this case, the SMP is also going to underestimate in high organic matter soils. So if you have something on a floodplain or you have higher organic matter, this might not be the best test. Adams-Evans, which University of Delaware runs, is good for soils with a low CEC and low organic matter. So what's that sound like? Low CEC can be a sandier soil, less clay, also less organic matter. So that fits coastal plain soils a lot better, and it's good for finding extremely small differences. So if you have a low CEC, it's a good way to find out what's a small difference to add lime, so it can be more accurate on soils that have lower CEC. But it may overestimate what we call pH-dependent acidity. The Malick single buffer is meant to reduce exchangeable acidity at about a pH of 5.5, to reduce the exchangeable aluminum that can cause toxicity to plants. It's best for soils that have a low CEC, but to me, it doesn't always consider crop needs. It might get rid of toxic aluminum, but does it consider that you might need a higher pH for soils or for crops that would prefer something between 6 and 7? So every single one of these tests, you can look up tables across the internet. Clemson has some for SMP. University of Delaware has all their tables for the Adams-Evans. Down here in the left-hand corner is one I use to talk somewhat about what Maryland would have done, where you have different textures. This clay soil requires more lime because it would have, obviously a clay soil would have a higher CEC, so it should have a higher buffer capacity. So you can go by the recommendation that comes with the lab report. You can simply look at it and know, but you can also look up these tables if you're thinking, I don't think this test fits my soil, and I would actually like to add more or less, and you can look up the tables that are used in some of these cases and try to do some adjustments in how much lime is added. So here's an example of the Adams-Evans table, and something else you have to keep in mind is what was the target pH. So simply looking at this table, you have over here on the left water pH, so that's that active pH. That's what I call the regular pH, water pH, measures your active acidity. Up at the top we have the buffer pH. So over here we have 795, that means the pH didn't drop much, so it didn't have a whole lot of reserve acidity. Over here it dropped to 7, so a soil that drops to 7 would have had more reserve acidity. Your water pH just indicates how acidic is the soil. Here it's 5.9 all the way down to 4.5. So if your water pH is 5.9 and you want to raise your pH to 6, it almost doesn't matter what your reserve acidity was unless it drops to about here. You don't need a whole lot of lime. They're not going to recommend it because if you're at 5.9 and you didn't have a lot of reserve acidity, there's no reason to add any lime. 5.9 is close enough to 6. If your pH was down around 4.5 and was very acidic, even though you might have had just a small drop in pH in the buffer, they're going to recommend a half a ton. If you have a very acidic soil and you have a lot of reserve acidity, this table is going to recommend up to 4 tons just to raise a pH of 4.5 to a pH of 6. So it's important, if you're looking at these tables, and sometimes people forget, that each table has been calibrated to a specific target pH, and the target pH is what pH do you want to raise it to. So you have your buffer pH, you have your lab test done, now what? Well, it might have a recommendation. It should be related to the crop type and your soil type, which you can follow. You can recommend that they add that much lime. But the actual target pH they might want to reach, or the test that was done might become important, and then you start to think about other considerations in the soil. Number one thing you can always discuss with anybody, pH of 7 it's a neutral pH, so everybody thinks, well, we need to get, the layman who hasn't had much soils, we need to get our pH to 7 so it's neutral and there's no acidity and we don't have any issues in our soil. But what they figured out back in the 60s and 70s is that actually we didn't need a pH of 7 because when we got above a pH of 5.5, aluminum becomes a solid. And if it's a solid, it can't affect roots. They figured out that aluminum was a main driver to reducing yields in crops as it was toxic to roots. Hydrogen isn't actually an issue until you get your pH less than 4.0, which is pretty rare to get in soils unless you might have some acid sulfate soils. So the main driver of reducing yields they found to be exchangeable aluminum that was free, floating around in the water, versus when you raise the pH above 5.5 and it becomes a solid mineral and isn't necessarily an issue anymore. So this is where that test, the Malick 3, comes from. At least if you get it above 5.5, aluminum's not toxic. Another thing to consider and keep in mind is that other metals like manganese and iron can also be toxic as your soil becomes more acidic, but they have to be in high enough concentrations. So if a soil doesn't have a whole lot of exchangeable acidity in the first place, you might not necessarily need to reach a pH of 5.5. So in some cases when you're having discussions with people, once they know it's 5.5, they think all soils are made the same. But if you don't have a lot of aluminum in your soil, you might be able to get away with a little less. Another thing to keep in mind is if you go to a pH of 7, and we'll discuss this in a minute, that you lose access to micronutrients, because just like aluminum and manganese and iron, as your pH rises, they can become a solid mineral. And plant roots aren't going to take up something that's a solid. They need it to be a dissolved ion. Something that doesn't come up a whole lot in this region, but if you happen to move to North Carolina or live there or in the Everglades, organic matter can bind aluminum and remove it from the soil. It can also bind manganese and other metals. So in North Carolina, they actually will say soils can go as low as a pH of 5.0 without any aluminum issues, and that you don't need to raise your pH to 5.5 in this case, because organic matter is giving you additional help in removing aluminum. But this is only going to be a factor if you know you have some soils that have higher organic matter. So this is where target pH becomes important to what crop are you going to grow. If we just focus on aluminum toxicity, this red line, if we just get to a pH of 5.5, yeah, we might have knocked out toxic aluminum. We might have reduced the amount of toxic manganese or iron if there was a lot in your soil. But is it still the best pH for your crop? And this table from Penn State shows you some of the better pH ranges for corn sit between 6 and 7. Alfalfa even higher at 6.5 to 7. Oats can actually do better about 5.5. So, and this question came up to me from somebody from North Carolina who did find that if they just followed that Malek test, if they only got to 5.5, they missed out on better yields, and they always saw better return if they raised it to 6.0. And these are decisions you can make from understanding your soil and not just going by the simple recommendation, but thinking about it a little harder. Target pH can also be related to phosphorus availability. Sometimes when I give this talk in this region, I forget to put this in because our phosphorus is so high. I don't consider pH to be something that's going to affect phosphorus availability to crops, but other parts of the state are going to see it. A pH of 6.5 is optimum for phosphorus. So if you know that phosphorus is limiting in your soils, if you maintain that pH of 6.5, it's going to be at its optimum availability, because as your pH drops here, it gets tied up more with iron and aluminum. Remember, as your pH drops below 5.5, these dissolve and release, so phosphorus gets tied up with them. Above this pH, you have more calcium, and phosphorus can get tied up with that. So in considering phosphorus, a pH of 6.5 is considered best. So again, the pH of 5.5 might get rid of aluminum, but maybe you need a pH of 6.5 to take advantage of more phosphorus. Micronutrients are also important. There are a whole lot of these diagrams on Google and on the internet. I usually use this one in my presentations because it already has a reference in it, so I won't get in trouble for stealing other people's work. So this one is from the Kentucky Agricultural Experiment Station. It's from 1970. A lot of these will show different ranges and different thicknesses, but what it gives you an idea of, your pH from here is 4.5 and rises to 8.0, and how thick these are determines how much is available, or say in the soil solution. So you see right here at 5.5, as you drop in pH, your aluminum picks up a lot. Same thing with iron and manganese. So as your pH drops, you can pick up in toxic levels of these. But as your pH rises, some of these micronutrients can have a lot less left in the soil. So that's a problem with raising pH too high, and this is another reason to pay attention to buffer capacity and the lime rate and the lime requirement, because it's a lot easier to add lime in increments, but once you push it too high and you lose access to micronutrients, you cause yourself a whole bunch of different problems. Predicting micronutrients in soils and their availability crops is tough. That pH of 5.5 for aluminum is a lot easier to discuss with people because we know in general you hit 5.5, you reduce the toxicity. Again, if your soil doesn't have a lot of aluminum, free aluminum, it might not be as much of an issue. But a pH of 5.5 is a good point to hit. It's a good talking point, but it doesn't fit all soil types. Soil type and texture may predict the micronutrient availability and toxicity. So if you have a clay soil that's high in CEC, it might hold more nutrients so you can push that pH higher versus a sandy soil, which might not hold as many nutrients in it. So you wouldn't recommend pushing the pH so high. If you overshoot your pH, you can easily get into this region where you don't have access to a lot of these micronutrients, and you're going to start to see crop deficiencies. This is a table, some work done in Clemson. It's coastal plain soils, and it's about manganese level needed for sufficiency for growing soybeans. So it's a very specific case and very specific soil types, but it gives you an idea of how much more of these nutrients you would need in a soil as your pH rises. So over here on the left, and I've got these outlined because I'll discuss a little later. University of Delaware recommends a pH of 6.0 for sandy soils and 6.5 for clay soils, and that partially has to do with you're more likely to have that amount of micronutrients in those soils. But as your soil pH, if it's at 5.5, you only need 3.5 to 4 pounds of manganese per acre to be sufficient for soybean growth in this study. By the time you get to a pH of 7, you need 17 pounds. So what this table is telling you is you need more total micronutrient in a soil as your pH rises just to make sure some is going to be available to your crop. And this is the same for zinc and iron and other micronutrients. Because in this case, the 17 is telling you you need a whole lot of manganese just to make sure some is going to be dissolved and available for your plants. So it's very easy to look at buffer capacity and lime requirement and just follow what's on there, but because of variation in soil types and availability to micronutrients, you have to think a little harder about it. But then there's also the flip side of that. If your pH is too low, we already know that you can have toxic levels of aluminum, but you can also have toxic levels of manganese. It's rare. To have toxic levels of manganese in your soil, you would have to have enough manganese there in the first place. It's the same thing with iron and aluminum. So you do have to consider the other side of lowering that pH. Down here on the bottom, you can see that if you see a soil that's colored in red, it's coated in iron and aluminum oxides. As that pH drops below 5.5, some of that's going to dissolve, and it's going to release iron and aluminum, which can reach toxic levels in your soil. But it's only a problem if they dissolve. So University of Delaware is going to recommend for a sandy soil a pH of about 6.0, and for a clay soil a pH of about 6.5. What you have to remember about these and anything in soils is it's just a general recommendation. It works well relatively. So a pH of 6.0 for a sandy soil is mostly due to it has a low CEC, so it doesn't hold many nutrients. Sandier soils are going to weather slowly and not release as many nutrients, and they should be low in micronutrients in the first place. So if they're low in all of this and they can't hold a lot, you can raise your pH. It's not going to have a lot of buffer capacity. And you're not going to have enough micronutrients, so if you raise it to 6.5, your crops might not have access to them. Versus a soil that has clay in it. Clay can hold more nutrients because of its CEC. Those clay particles might dissolve and weather faster, and if they have nutrients, micronutrients in there, they can release them. And often these soils, particularly when you're talking about soils formed from bedrock, should be greater in micronutrients depending on the bedrock they form from. The problem with these recommendations is they're just general, so you can't take it for granted. Some soils might be so low in micronutrients that you don't want to go above 5.5. And if you have low aluminum, maybe they can push it a little lower. So don't assume, just because the recommendation is 6.0, that if you give that recommendation to every single farmer that has a sandy soil, you'll get the best result. In some cases, you might actually cause a deficiency. So this is a picture of a field that hadn't been limed in about 6 years and received lime. One point about this field to discuss is soil tillage and mixing. It's a no-till field. So if you add lime to a soil that isn't tilled, where the lime isn't mixed, if the lime isn't mixed in the soil, you can actually have a differentiation of pH from the surface down to the subsurface. And this is an extreme example, going from 7 to 6. So if you're at a pH of 7 in the surface where you're planting these seeds, your crops can quickly have a deficiency in micronutrients. There are cases where if the roots get beneath this point, they can get access to micronutrients where the pH is lower. So it might only show up early in the season and then your roots get down and everything's fine, or you can end up like this soybean crop where you can see the upper 2 inches had a pH of about 7, 2 to 4 it was 6.9. So the pH was too high for this sandy soil, and he had a deficiency across the entire field. And part of this is going to be due to if it's not mixed in well. It's one reason you're going to recommend to people that you put stuff out in the fall, you put stuff out in the winter, and you give it time, especially in a no-till soil, for rainfall to work the lime down in here. Now, if you go through here and disc the soil, you can mix it to 6 inches a lot quicker. And you certainly wouldn't recommend to anybody to put lime out the day before they're planting. So what was interesting about this soil, we went out and took some leaf tissue samples and some soil samples to see what was a deficiency, to see if we could actually discuss the manganese and pinpoint it. So it's what you'd expect. The leaves looked like they had a manganese deficiency. The soil showed that it was deficient in manganese, and so did the leaf tissue. So this made sense. We had a pH of 7. Our soil was already low in manganese, so was the crop tissue. It had no manganese it could pull up. What was also interesting, though, is that the leaf was deficient in copper, but the soil was not. So in this case, what we saw was the soil had plenty of copper that could be extracted in the lab, but the soybean had no access to it, because in this case, the leaf couldn't pull it up. So this was a better example that your soil can actually have plenty of the nutrient there, the micronutrient, but if you raise the pH too high, there's no access. The manganese, on the other hand, which is a little more common on our soils on the coastal plain, our soils are already low in manganese. And there are some farmers over here that I've talked to who are afraid to push their pH above 5.2 to 5.5 because they think they already see manganese deficiencies. But something to consider in this case when making any kind of decision in agriculture is there's a lot of ways to approach this. You could keep your pH lower if you wanted to make sure your crop had access to what manganese was there. You can also obviously add manganese to the soil. But another thing you can consider late in the season is to go across and add a foliar application of manganese directly to the leaves. So there are decisions and choices you can make. It's not as simple as like, well, we have to get it to 6.5 because that's best for corn. We have to get it above 5.5 because we get rid of toxic aluminum. You can look at all of this. Do I have enough aluminum to be worried about? Do I have too much manganese so it might be toxic if the pH drops? There's a lot of different factors that you can think about and discuss when raising the pH. You don't have to just go with one prescription. This is what a copper deficiency looks like. It's a little hard to find pictures of these because a copper deficiency isn't very common in this region, but in this case, his pH on the sandy soil was just raised a little bit too high. These were leaf tissue samples that were taken from the soybean plants in that field. What was interesting is this farmer was actually told that this was a disease or bacterial infection, partially because they saw some of the necrosis on these leaves and just thought it was simply an infection breaking them down. But actually the chlorosis that you see along these veins gives you an idea that it's a nutrient deficiency. But even along the soybean, we had a range in the deficiency, and you can see some of these spots that kind of look like what is back here is the copper deficiency. And it's not something, manganese, because it's common on the shore, because it's common on our soils, it's easy to spot pretty quick. We're going to look for it and say, well, that's probably manganese. But it's always a good idea if you see a deficiency to do a soil test. In this case, the soybean field was too far gone to do anything about it, but now he knows next year that he's going to have a problem with manganese and copper, so he can either try to lower the pH, or he can make sure at a certain time of the year he either adds more of these micronutrients into the soil with his fertilizer or as a foliar application. So determining if you have a lime issue in your fields can be obvious if you have an issue, but exactly what's wrong with it is not simple to figure out unless, to me, you do a tissue test to be sure what's in there, because we wouldn't have known that there was an issue with copper until we did the test, because the soil showed plenty. So in this case, taking a tissue and soil sample together to make sure you know, because we could have come up and said, well, there's a copper deficiency, the soil needs more copper, and in this case, the soil had plenty of copper. It was just the pH was too high. So anybody that's going out and adding lime, especially to a field like this that hadn't been limed in 6 years, scouting your fields is very important. You don't just add the lime and assume, well, I followed the recommendation, so everything's okay. In this case, the soil was low enough in these nutrients that it might have raised the pH, but it raised it too much, and he wasn't aware of the buffer capacity of the soil and quickly overshot what the pH should have been. Another thing to keep in mind in using the internet to figure out if you have deficiencies, that a lot of them can end up looking alike. Now, we don't really have iron deficiencies in this region, but they do have them further out west. But if you look at how they describe manganese and iron, they almost have the same exact description, even though they don't point it out when you read it. Interveinal chlorosis on the upper leaves, they describe manganese and iron as both way, dark green veins, occurs on high pH soils. So it's not just helpful to look at pictures. This is why a tissue test. This is a picture in Nebraska of manganese deficiency. This is what iron deficiency looks like. If you see enough of both of them, you might be an expert. Now again, coastal plain, we're more likely to have this than this, but it doesn't mean there can't be a case out there somewhere. Same thing here with all these pictures. Everything on the left is manganese. Everything on the right is iron. And if you're not trained in it, if you don't do it all the time, they can look pretty similar, which is why in most all these cases you should recommend to do a tissue test, because you want to know what exactly was what wrong so you know how to solve the problem. So when evaluating the lime requirement, some of the things that you want to keep in mind is that all those lab tests are not equal, and we have a multitude of labs that we can pull from, and all these labs might have different calibrations that they've done to figure out what the lime rate is. You can easily follow the lime rate that's given, but in that case, the farmer, the producer, is going to want to go out and scout and make sure it worked. You don't have to do a soil test every time. You can have a pH meter, you can have litmus tests just to make sure the soil reached the pH that you wanted it to and it didn't overshoot it. But if you use different labs every year for the same fields, if you have different soil types, but you know one lab is running something that's not doesn't fit the soil type you have the best, you can consider raising or lowering the amount of lime you add. You don't have to just go by the recommendation. You can learn your soils better, and you can make decisions on whether you want to increase or decrease that. So understanding that soils have a varying amounts of acidity, especially, even though you might get used to soils in your region, when you discuss soils with people across the state and the country, we don't all have the same problems. And if we don't understand that, we can confuse each other. Buffer capacity is going to vary, the amount of organic matter is going to vary, the amount of micronutrients in those soils can vary, even across a small space. So knowing this can give us better prescriptions for fields. And always, if you've limed, scout the fields. It can be as simple as a pH tester out there, but making sure, walking out and looking at these crops and knowing the difference between, is it a disease or is it a micronutrient deficiency, and then talking to consultants, talking to ag agents to make sure you know what the problem is so that the next year it can be solved. And if you catch it early enough and it's a micronutrient deficiency, there can still be foliar applications to these leaves. And then, of course, you always want to give that lime plenty of time to react, especially as we turn more to no-till in the region to improve our soil quality and health and reduce erosion. But you want to make sure that lime has plenty of time to react, plenty of time to reduce the acidity you want it to, because some of these limes, when we add them, they have to react with the acidity, so they can be sitting around in the soil for a while, and they can maintain the pH higher than you want it to be. So these recommendations are meant to, say, raise the pH of the upper 6 inches of the soil, but it's not going to do it immediately. So you have to think about mixing with the soil and giving it time to raise the pH where you want to and react with all the acidity in the soil. Okay, that's about all I had for this talk, so if there are any questions.